high-throughput neuroanatomy, now called connectomics, as well as optimization approaches to understand brain structure, function relationships. But in the last couple of years, I kind of returned to my first love, and we made some progress on understanding visual processing. And it turns out that the principles that we used were around for a while, and they were actually inspired by engineering. So in today's talk, I'll try to cover a little bit of what has been known before about visual processing, and I will slowly move to newer and unpublished stuff. So the whole talk, as you can see, will be devoted to visual processing in vertebrates and invertebrates. And here are the heroes of the talk, today's talk. And I will focus on the two questions. One is the shape of spatio-temporal receptive fields in early visual system in both vertebrates and invertebrates. And again, some of that stuff has been published before. We just revisited it. And the second one is more focused on the circuits and how those receptive fields arise mechanistically. And because this is kind of an interdisciplinary talk, I welcome interruptions and questions. So if there is anything you want to ask, please interrupt me. I intentionally planned the talk to be shorter than the time slot I have. All right. So about 60 years ago, scientists like Haldane and Koffler made a huge advance in understanding visual processing by figuring out how to record from the neurons in the visual system. So as you know, the way we see the world is that the image is formed on the back of the eye on the thin layer of neural tissue called the retina. And photoreceptors convert the light into the electrical signals that they pass on to the bipolar cells, which in turn pass it on to the ganglion cells that project to the rest of the brain. And what Koffler did, he learned how to electrically record from the ganglion cells, thereby eavesdropping on the communication between the retina and the rest of the brain. And here's what he saw. If he presented a dark screen that is nothing to the eye and listened to the neuronal firing, this indicate individual spikes, but for the purposes of this talk, we will not be interested in spike timing. We'll just characterize neuronal activity by the firing rate, which is the number of spikes per second. And that's shown by this red line. There is some low spontaneous rate of firing that he observes. Then, when he showed a light spot centered on the location of the ganglion cell that he recorded from, he saw a response which had this transient component, but also this steady state component, which ended when the stimulus was withdrawn. So this was the basic experiment, not entirely unexpected, but the interesting observation was that the response had this major transient component. And this has been repeated in many other experiments in the visual and other sensory systems. But then something interesting happened. When Koffler showed a wider spot of light, he noticed that the response was much weaker, even though the stimulus seemingly was stronger. He hypothesized that this happened because the region surrounding the narrow spot of light, this annular region, has an inhibitory effect on the neuron. And to support this hypothesis, he presented an annular stimulus, and indeed, that stimulus just suppressed spontaneous rate of firing in the neuron, leading to a rebound after the stimulus was extinguished. So these two observations, the transient response to a stimulus and this interesting shape of neuronal activation, which is usually called a center surround receptive field, formed the basis of our understanding of retinal processing. To understand the center-surround receptive fields, what you can do, you can think that the retina applies a filter on the incoming image, which has positive components in the central part corresponding to that narrow spot of light, and negative components in this annulus surrounding that narrow spot of light. Then if you filter a natural image like this, and the reason I chose this image is because I work at Janelia Farm, and people think that this is a natural image for us. It's not really true. We're in suburban Washington. It's a heavily built up area, but just to maintain the legend, I put it in. Okay, so if you apply this filter onto the image, you get something like this, where you emphasize edges of the objects as opposed to uniform, to regions of uniform intensity. Okay? So the basic observations of early visual processing then is that ganglion cells act as edge detectors, both in time due to their transient response and in space due to their center-surround receptive fields. And the question we would like to ask is, why is that the case? If you open a textbook, it will tell you, well, that kind of makes sense, because what the visual system wants to know is it wants to parse the visual scene into objects. And so to find the boundary of objects, you need edge detectors. And so retina kind of tells the brain what the brain wants to hear. And this may very well be true, but it doesn't help you to build a theory of what's going on. For example, to derive those receptive fields. Well, interestingly, about the same time that Kufler was recording from those ganglion cells, another way of thinking about the system has emerged. And the idea was to think about the visual system from the engineering perspective and understand how it has been adapted in the course of evolution to transmit information about the world to the brain. And the key observation here is that if you look at a natural movie like this one and plot the intensity of this one pixel as a function of time and look at the intensity variation, you will find out that the dynamic range of intensity variations is huge. So if you go out of this room outside, the variation in light intensity will be more than 5 orders of magnitude. But even if you stay in this room and you kind of just look around, then the variations of intensity are at least 3 orders of magnitude. Moreover, it is known psychophysically that your visual system can detect about 3 orders of magnitude of intensity gradations. But that's rather interesting, because if you think about a single neuron like that ganglion cell that Kufler recorded from, its dynamic range is much smaller, perhaps just 2 orders of magnitude, and that's optimistic. How do you arrive to that number? Well, think about a ganglion cell having firing rates which could vary from 1 Hertz to 100 Hertz. And let's give it 1 second to make a call. Then there are about 100 levels that it can use. It's still much smaller than the dynamic range that it is faced with in the natural world. So there is a major mismatch between the dynamic range of the sensory stimuli in the world and the dynamic range of the ganglion cell that transmits that information. And that led scientists like Otnev and Barlow to propose ideas having to do with the compression of the natural stimuli by those cells using so-called redundancy reduction. By just transmitting information they absolutely have to transmit to encode the stimulus. And this work led to the whole direction in systems neuroscience, looking at the normative, using normative approaches and so-called natural image statistics and understanding the properties of visual and other sensory systems. But the ideas of Otnev and Barlow themselves were taken from engineering, because about that same time, engineers were developing long-distance communications networks, like phone and otherwise, and they developed compression algorithms that actually inspired them to say that. So let me give you a little bit of a review of signal processing in relation to this problem, and then we'll come back to neuroscience. Are there any questions at this point? Okay. So in signal processing, the family of compression algorithms that are used for this purpose are called predictive coding. And the idea of predictive coding is if you have a signal with large dynamic range, which is highly correlated, you can actually compress it by encoding only the parts of the signals that you could not have predicted from the previous values of the signal. That's why it's called predictive coding. So let's give an example. Just for simplicity, for this illustration, let's consider discrete signal. Let's say varies from -10 to 10, and discrete time so that I can actually plot something. And suppose we want to communicate this signal with a large dynamic range through a communication channel that has only 3 values, -1, 0, and 1. If the signal is changing slowly, like the one here, you can actually do that by building a circuit of this sort. You split the incoming signal and send this path to a predictor, which is trying to guess the next value of the signal. It subtracts its prediction from the actual incoming value, and what is being transmitted is not the signal, incoming signal itself, neither the prediction, but the difference between the two, or prediction error. Then if your prediction is good, the error is small, and then you reduce the dynamic range of the signal. So the simplest predictor one can come up with is to say, well, the signal at the next point of time is the same as the day before, like for the weather. Or here, the same as the time point before. So I can show it on the cartoon by copying this original signal and offsetting it by 1 step in time. And then I would implement subtraction as shown here, and this is the prediction error that I would get out of it. So because the original signal varied slowly, what happened is that the dynamic range was reduced as a part of this operation, and you can squeeze it through this limited dynamic range channel. Okay? Another way to describe this encoding is by considering an operation of this prediction error filter on the original signal. What it does, it takes the current value of the signal with amplitude 1, this is this line, and then subtracts the previous value of the signal with the weight -1, and this is this line. Okay? But we will use this convention a lot from now on, so I wanted to introduce it now. And I also want to point out that here you go back into the past in time. These are previous values of the signal further and further back in time. And the reason people reverse the time convention is to make it consistent with impulse response, which we will use to test these predictions. Okay? So you can look at this filter and you can see that it is kind of a discrete derivative, right? You take the current value of the signal minus the previous value of the signal. So when you want to invert it on the receiving side, the opposite of the derivative is an integration, and that's why you build the circuit which effectively integrates the signal, and you can reconstruct it by putting it through the circuit, and that reconstruction is in fact lossless if you don't have noise in the transmission. So this is indeed a compression scheme that works very well. Each one of you is a proud owner of such device because it's used as a part of GSM cell phone encoding and so on. In this particular example for this natural video that I showed you, when you do this kind of compression, you reduce the power about 6-fold. And by the power, I mean the mean squared error of the signal, of the prediction, which is what we'll use to quantify the quality of our compression. In application to the brain, I will use only the front part of the scheme. And I don't want to imply that the brain is trying to reconstruct the signal on the receiving side. The only reason it's here is to prove to you that this compression is lossless, that no information gets actually lost. But I don't think it's necessary to assume that the rest of the brain is trying to assemble the stimulus back, because the goal of the brain is not to represent the stimulus, but to act on it. And there is no reason to believe that it's easier to learn how to act on the stimulus based on the original representation of the stimulus rather than on compressed one. Okay? But we will use this as a model of the early visual system. Any questions? All right. By using this very simple predictor such as delay, we were able to reduce the dynamic range 6-fold. Can we do better than that? And in fact, one can. And if you restrict your predictor to the family of linear predictors, this problem is easily solved. This is known from signal processing. You can come up with an optimal predictor, linear predictor, which is a linear combination of the past values of the signal from the previous time point, 2 steps before, 3 steps before, and so on, with different weights. And then the transmitted signal is the difference between that prediction and the actual value. And you can find those coefficients W by looking for a minimum of this square of the prediction error averaged over a stimulus ensemble. So this is an important point. The optimal coefficients W depend on the signal ensemble that you're working with. Okay? So in this case, for our natural movie that I showed you, you can compute those W's by solving this as quadratic functions, so you can solve it analytically, actually. And you can find an optimal prediction filter that looks like this. As you can see, it still has a coefficient of 1 corresponding to the current value of the signal. This is this line. But then the coefficients for the past value of the signals are much less than 1. And actually, all of them summed together are also much less than 1. But the prediction now depends not only on the previous value of the signal, but also some of the older values. And the reason you would do that is if there is some noise in the signal and you want to come up with a better prediction by averaging it out. Please.
But how is this better? Because if I just have to transmit -1, 0, and 1, I need much less bandwidth. I'm going to have to now transmit this very fine variation. So it's not just about the range, but also the resolution of the signal that you should care about. And what you now introduce is actually I have still a smaller range, but then the values I have to transmit have a very small variation that I have somehow to take care of.
Yeah, so that's a very good point. So the example that I use uses discrete transmission, but this optimization is done for the continuous signal, for analog signal representation. So if you want to formulate this problem in a full-fledged way, what you have to do is to say that there is noise in the communication channel, and that noise contaminates the signal. And then you want to transmit the signal whose amplitude is as big as possible relative to noise, but within the dynamic range of the channel. And so what you do, you try to reduce the dynamic range relative to the original signal. That's what counts. And then we would have some sort of a gain that comes before the actual communication channel, which will scale up the signal relative to the noise that will be added. So that's where your small variations will get amplified relative to the contaminated noise and transmission.
Okay.
Does that answer your question? So I wanted to avoid all those issues in the example that I gave you initially by discretizing the signal at values which exceed the amplitude of noise. So you don't have to worry about it. But in reality, yes, of course, there is transmission noise, and that's what happens.
So Dmitri, sorry, we also worked on predictive coding, as you probably know.
It's in the next slide.
But there might be, this system takes away the DC component, doesn't it?
Yeah.
So, but there are other parts of the visual pathway that do rely on the DC component. For example, the thing that controls the pupil will need DC information. So would you say there's a parallel pathway that
I think so.
does that?
I think so, but the bandwidth is much smaller there.
Okay.
The other part of the answer could be that in practical implementations of predictive coding, especially when there is communication noise, the way people deal with it is that the subtraction is not done perfectly, and you inject a little bit of a leftover of the original signal in the transmission. And that actually, and if you reconstruct it with the leaky integrator instead, then that actually allows you to overcome some effects of noise, but also keep track of the background level of the signal. Okay? Any other questions? All right. So then what we have is the prediction for the optimal filter that when applied to the original stimulus, which is in black, gives you a prediction in blue, and the error has a much smaller dynamic range as advertised. How does that correspond to neurons? Well, what we're going to do is to compare the prediction for the shape of that prediction error filter with the response function of neurons. And the reason that makes sense is the following. So let's think about what the predictive coding system does. This is this time series, the input time series, the natural signal that we're trying to transmit. This is the value at time t. This is the value at time t minus 1, and so on. And as we agreed, the current value at time t is transmitted directly with coefficient 1, but the previous time value is multiplied by coefficient w1 and subtracted from the current value along with other previous value. And so what we're going to do is trying to test experimentally what those coefficients w are. And the way we can do it is by looking at the impulse response. What it is you present a stimulus with a delta functional, you present a delta functional stimulus to the system, like a flash of light, which is this red square, say, at time point t. And that flash of light, of course, is transmitted with unit coefficient, so you're just going to have a spike. But if nothing else is presented afterwards, at the next point of time, you should think of that flash as occurring one step before. And therefore, the response you would see at that time point will be that flash of light times coefficient w1. At the next point of time, the flash goes back in time even more, and then you're probing coefficient w2, w3, and so on. And this way, you can test the prediction for your optimal linear filter like this one by recording electrically from the neurons in response to a light flash. And this is what has been done by Mandiam and collaborators more than, let's see, 30 years ago in fly, in an invertebrate, where they recorded from second-order neurons called LMCs that actually come in two kinds, L1 and L2, which will become important in the second part of the talk. But they receive synaptic inputs from the photoreceptors, which transduce light into electrical signals. And when they recorded from those neurons in response to the light stimulus here, they saw this biphasic response. And they argued that this should be understood as a measurement of the impulse response of a system which could describe as an optimal linear encoder of the incoming information. And this is the optimal linear filter they computed for high signal-to-noise ratio. So biphasic response is nice, of course, but they didn't stop at that. To make their case more convincing, they decided to vary the ensemble of the stimulus. Remember, the optimal linear filter was computed for a particular stimulus ensemble. So if we change the stimulus ensemble and we believe in this framework, then the filter should change as well. And that's what they did by altering the intensity of the background light level. They effectively went into a regime where signal-to-noise ratio was much smaller than before. And then the optimal linear filter that they computed had a lower amplitude but longer predictive component. The reason why that happened is because in lower signal-to-noise ratio, to come up with the better prediction, which is less contaminated by external noise, you would want to average over many steps in time. And when they measured response of this LMC cell, they found that, in fact, that second phase of the response had lower amplitude and longer extent, just as one would have predicted from the linear predictive coding framework. Was there a question?
Yeah, so because I don't really understand your rationale, because on the one side, you have the optimization of the linear filter, that I suppose I would say something about the optimization has a time scale that's a little bit longer than just changing the stimuli. So the change, and so what you say is that I see a difference in the linear filter has to do that I change my weights of this linear filter. But you could also just say, well, the retina is not linear. So this means that when I put in a combined signal, I get out a different response because I have a different input and does not sum up linearly. So my point is they see this difference because it's a nonlinear filter, and somehow what I understand you suggesting is that we change the linear filter in this short.
Right. So this work was a rather convincing evidence in favor of the predictive coding hypothesis, I would say, because not only you could account for the biphasic shape of the response, but you could also predict the change in the filter as a function of the change in the natural stimulus ensemble. But if you look a little bit more carefully at those curves, and I'm actually interested as Wendy would agree with me, then there is a little bit of a discrepancy between this observation and this prediction, because the first phase of the response is not infinitesimally short as you would expect when this is a delta function or just a single time value with coefficient 1, but it has a finite extent. And you can say, well, of course, the brain doesn't work in discrete time. It works in continuous time, so there must be some spread. There is no real delta functions in nature. But if you look carefully, the width of the peak actually increases, so it changes depending on the signal-to-noise ratio. So there is something else going on. And I think we can guess what it is. And this has to do with the fact that the incoming that the predictive coding system, the way it was described, works well only if the stimulus has strong correlations and changes very slowly. If you get a stimulus that is uncorrelated with fast, unpredictable changes, the system doesn't work well. Because consider, for example, the case when you get white noise input to the system, and then it's trying to predict, so it's taking the previous value, but what you actually get is uncorrelated with that prediction. Then what will happen is that it will actually increase the dynamic range of the transmitted signal. Why should we think about such kind of stimulus? Well, for example, because the real signals are usually coming with a lot of noise components, and the noise presumably is uncorrelated. And so at low signal-to-noise ratios, what is going to happen is that the dynamic range of the signal will not actually be reduced by the circuit, but it could actually increase. And so to fight that problem, what you can do is you can put another linear filter in front of the predictive coding filter, which denoises the signal by, for example, low-pass filtering it through this kind of shape. Then you effectively take out this noise first and then encode the signal with the predictive coding framework, and the actual filter that is used to describe the system will have a finite spread of the first phase of the response. And that is, in fact, the explanation that Srinivasan et al. Used. They said, OK, we know that photoreceptors implement low-pass filtering from electrical recordings directly from the photoreceptor. And so by convolving those two filters, we can account for the width of the first phase. And their theoretical prediction, in fact, is that dashed line. Of course, what is assumed in this framework is that the system somehow knows the magnitude of the noise, and the statistics of the noise, as opposed to the signal, to figure out what that linear filter is. In the situation that was used experimentally here, this is indeed the fact, because the way they change the signal-to-noise ratio is by playing with light intensity. And the reason, the major component of the noise in their experiment was the photon shot noise. Just because at the lower time levels, you cannot forget about the discrete nature of light. The count of photons is integer, and so that introduces noise to the relative continuous value that is determined by the reflection coefficient of the objects. And that is the kind of noise, the amplitude of which you can figure out from the intensity value. So you can, in fact, know what it is. And then you can construct an optimal denoising filter, which you then convolve with the predictive coding system. And that is what we have done. So as opposed to taking a set, measured, low-pass filter response of the photoreceptors, we just computed an optimal denoising filter and convolved it with the predictive coding filter. OK? And then you can account for this response in flies. All right? Is that clear? OK. So now what I said in the introduction is that if we have general principles, they should apply across species. So let's see if this works in vertebrates as well as in invertebrates. And for that, I want to briefly put up the description of our visual system that I'm sure you know. So far, in the introduction, I talked about Koffler recording from the retina. But then the visual pathway takes you to this kind of relay station, as it's called, which is LGN. And then information is transmitted to the primary visual cortex. And in thinking about comparing this normative predictive coding theory to experiment, we have to decide which recordings we should be looking at. Of course, we can look at the recordings from the retina, but I think it's even better to look at recordings in LGN. And the reason is because if we are thinking about the optimal linear filter, it may actually describe the operation of the system that happened in several stages. And so you want to look as far as possible within the system where you still think that it does some sort of processing described by linear filtering. We know for sure that the primary visual cortex involves nonlinear operations because you cannot describe complex cells linearly. But LGN is known to have properties which can be largely understood within the linear filtering framework. And so what we are going to do is to compare the predictions of the linear filtering theory with the recordings in the LGN. In the vertebrate system, it's difficult to probe the response by just presenting a flash of light. And so people usually use another way, which is called reverse correlation. Just to make sure we're on the same page, here's how it works. You present a time series of intensity, for example, and you record spike in response of a neuron in the LGN. And then to figure out the linear filter, what you do, you average light intensity in the windows preceding each spike. If your stimulus is not white noise, you also have to decorrelate to reflect correlation, say, in natural scenes. But this is basically how you get the filter. And when these fellows did that, this is the temporal receptive field that they recorded, which we can now compare with the optimal linear filter computed for predictive coding. As you can see, it has the same shape as before, where there is biphasic response with the second phase smaller than the first. So I would argue that the predictions of the predictive coding theory apply across species, and there are examples of it working both in invertebrate and vertebrates.
So have people looked at the actual spectral shapes of the input signal and shown that the spectral shape can be accounted for by looking at the precise shapes of the filters? To do that, you'd have to change not just the gain of the input signal like you were showing with respect to signal and noise, but in fact change the shape of the spectrum of the input signal.
Yes, there has been some work in that regard that again has to do with the adaptation of the system to the natural stimulus statistics, and you can vary it somewhat. I'll show a little bit of examples of that. But I think there is a research question that can now be investigated more fully about how the system adapts to such variations, which is very interesting.
Yeah, so to follow up on that, the problem I'm having, I'm not sure it's the right point in time to pose it, but you know, if you look at this retinal LGN system, now from this perspective of coding, there's in some sense already a very big difference as compared to the engineering system we're looking at. Because in our engineering systems, the transmission was an issue. We always had bandwidth issues, right? From retina to LGN, actually, this bandwidth issue doesn't seem to be so large. You have a massive number of fibers going directly. So the issue of compression might be a somewhat different one. And this is maybe also evidenced in this temporal receptive field. So because you could argue, and of course I would like you to help me understand this better, neurons do show already a non-specific after hyperpolarization, right? So that means that the negative component that you interpret as being now reflecting this bipolar aspect of your filter might be just a non-specific aspect of neural firing, of biophysics, that you would find in any neuron, whether they're encoding some signal or not. So this is one issue. So does the brain, or does this retina LGN system solve indeed the same encoding, decoding problem? And isn't then the after hyperpolarization not an alternative explanation for the negative part of your filter?
So let me break this into two questions. The first one, we kind of motivated the predictive coding framework by saying that there is a bandwidth or rather dynamic range limitation, but you're saying, how do we really know it's there? And I think in the retina to LGN transmission, one can kind of make this argument because, yes, you could have the more fibers, but the thickness of the optic nerve is already, I think, about 1 centimeter, as it is in us. It's like as thick as a pencil. Having a thicker one might be detrimental. That said, this argument is much harder to apply in other systems, like the visual system of the fly, for example, where the bottleneck is not clear at all. So while we started this predictive coding, thinking about this predictive coding framework from that perspective of the bottleneck, we are now shifting away from it, and we think that this decorrelation of the stimulus, it has an important computational role, not just in terms of compression. Okay, but we can discuss it after the talk.
Is this related to what are called matched filters in detection theory, where you're trying to see signal and noise? I don't know what that is.
I think it's, yeah, but it's slightly different, I think. Sorry if I may comment.
Yeah, please.
It's a related concept, but I think it's slightly different. In the matched filter, you're looking at the spectrum of the object you're trying to detect, and you're trying to tailor the filtering of your system to match the spectrum of that object. So your system produces a maximum signal for that object compared to everything else that's around. So the impulse response that you would get in a matched filter would be quite different from this. This does not assume any prior information about what the object is going to be. It just has general information about the power spectrum of the scene, right?
Yeah.
It says, given that, what's the best way to encode the system with minimum redundancy?
The match filter is the denoiser that you were talking about before.
Okay, yeah, so there is no information here about what kind of signal you're expecting. Nothing about the function of the visual system other than you want to get as much information as possible. Okay? Now, the second part of Paul's question, which is also very good, is, well, how do we know this is really predictive coding? You know, couldn't be just biophysics or something else, right? There is always this possibility. And I think generally in science, you can never prove that your theory is correct. You can only disprove theories. The only hope that I have is to show how many things the single framework can predict that are then seen in experiment. And so what I hope will happen is that I present more and more predictions coming out of this same framework. And then let's see if you will still think that it's a biophysical effect. Okay?
But maybe what you have to argue then in this case, that there's a decoding of this component at the level of LGN. That possibly also then is reflected again at, let's say, a next stage of processing in V1, right? And that is not so obvious.
Yeah, I'll get to that in a couple of minutes. So,
but in fact, every filter will have to be implemented using biophysics. So the fact that there is a biophysical mechanism that produces this negative after polarization is not an argument against it being used as a predictive filter, right? So
it could be non-specific.
Right.
You have to show it's specific.
It could be just there because the system couldn't work otherwise.
Exactly.
But couldn't work otherwise, as in.
Once you emit a spike, you have to have polarize.
That's it.
But it varies in interesting ways when you vary the mean luminance, right? In predictable ways.
Exactly.
You probably wouldn't be able to predict just in terms of pure after hyperpolarization.
Sure.
It could still be a coincidence.
Could still be a coincidence, yes. But
could be a useful coincidence. It's something that the system found. Yeah.
It's the right time. It's the right time to tickle me, because what I was going to say is that this really? I was a question to the students. Is really enough evidence to think that this is predictive coding? Okay, and what I was going to do is to show you more evidence. Okay. So far, we talked just about the transient part of the response in this Koffler experiments. Okay. Okay.
There is a point. He has a point.
No, it's not really emitting anything.
Oh, that's much better. Thank you. Okay. So this biphasic response, of course, accounts for this transient response, because if you apply this biphasic filter to the step stimulus, you would get this response. Okay? But there was a second part. But now this doesn't turn the slides. Okay. Wrong one. Right one. Okay, but there was the second part of Koffler experiments, which is the spatial receptive fields that have the center surround shape. Can we understand them starting with the same predictive coding framework? And by the way, I should mention that starting with the seminal work of Srinivasan, Laughlin, and Dobbs, other people have developed this, such as Attic and Van Hattering, and in slightly different context, Rall and Bard. And these are the other esteemed speakers at this school. So keep that in mind. Okay, so can we account for the spatial receptive field? Okay, when we think about the spatial receptive fields, we cannot think about a single communication channel anymore, or a single photoreceptor or a single ganglion cell. Now we have to think about a group of photoreceptors transmitting through a group of ganglion cells through multiple channels, and then we have to encode this multi-channel signal in the efficient way using the predictive coding framework. Can we do that? In fact, we can. And this is the scheme that is the generalization of the one I showed before to the multi-channel case, where instead of having a single channel, where these are the values of the signal in time going back and past, you have multiple channels corresponding to spatially adjacent ganglion cells. And then if your goal is to transmit this signal with the least dynamic range, you again subtract a prediction for that value of the signal. But to make that prediction, you don't just use the values of that signal in the past points in time, but you also use the past values of the signal from the adjacent channels or adjacent photoreceptors and adjacent ganglion cells. And so the signal is now vectorial, and so are the coefficients W. Each of them, W1, W2, W3, and so on, are vectors. We can, of course, handle this with linear algebra, and we can compute optimal linear predictive vectors and see how it works. When we do that, it turns out that the transmitted power reduces even more, down to about 8% for the natural movie that I showed you before. And we can also compute the optimal linear filter that we are going then to compare with the electrophysiological measurements and see if that can convince Paul. Okay? So how exactly did we compute those filters? Just to go through a few steps to make sure there is no magic here. We take a natural movie, exactly the one that was used to stimulate the cat, from which the recordings were made. We add noise to that movie. This is just additive white Gaussian noise. And then we compute the optimal denoising filter by minimizing the error between the output of that filter and the original clean signal. Again, we're using the assumption that this is a situation where the nature of noise is known. For example, it's photon shot noise, which scales known wave with intensity.
This is the Wiener filter.
Yes, it's just, that's very, thanks. This is a Wiener filter. We do not invoke the condition of causality here because there is a long delay in photoreceptors, and so the signal kind of hangs out there for a while, and that's why I think we can use a non-causal filter, but that's not essential. But the filter now is not one-dimensional in time. It's actually a three-dimensional filter because there is one time component and two space components, x and y. So the filter is three-dimensional. I cannot draw you things in 3 dimensions, so what I will draw is a cross-section of the filter in time-space plane. And this Gaussian-looking blob is the optimal filter for this movie with the white Gaussian noise. Once that signal got denoised, you can put it through a predictive coding filter, which also is computed through the Wiener framework, minimizing the squared error. And it also has a delta function at 0 time, and then a predictive component further back in the past. But unlike before, it actually has a spatial component because the stimulus has correlations not just in time, but also in space, and you can use them to make prediction about the value of the signal and subtract and have a better prediction that minimizes the squared error. So it has this spread. And then what you transmit is the original signal minus the correlated parts of it that are taken out. So it's a decorrelated, a whitened version of the original signal that is being sent downwards. Because these are linear filters, the combination of the two is just a convolution. You can compute it by a simple linear operation in MATLAB, for example, and this is the filter that you get. So as before, it has a biphasic structure in time, but now it also has a structure in space. Now this may look a little bit unfamiliar to you if you haven't worked on this before, but this is actually the filter that Koffler saw. And to prove that, what I'm going to do is show how the spatial, how the filter looks in the spatial plane sectioning at this point in time. This is what it looks like. This is the center surround filter that was seen by Koffler. But what we get here is its full spatio-temporal form, not just the spatial part or just the temporal part. Okay? But there is no magic here. There is a unique result. You give me a natural movie and tell me the signal-to-noise ratio. There is a unique answer for the optimal linear filter that optimizes the way I described.
This is really lovely, and this is really beautiful. And I was going to say, maybe you've already done this before, but if you now expose cats to signals with different signal-to-noise ratios, would their filters adapt?
Yes, that will be shown on the next slide. Thank you.
And zero, zero here is the response of the readout neuron, or the stimulus onset.
Yeah, this is the stimulus onset. See, in the retina, there is a huge delay in the photoreceptor, like 60 or 70 milliseconds. And so that's why there is this offset, and there is no requirement, I think, on causality on the denoising stage.
In fact, if I were to think about the downstream stuff a little bit, there is one thing one could add to this picture, which is supposing I have a noisy channel that follows my encoding. In fact, I may not want to encode more than I can transmit through that channel. I wonder if you have thought about that.
Right. So let me go back to that question where I discuss comparison with Attic and Van Hatteren, because that's the kind of point of view that they chose to attack this.
But now, wouldn't the prediction of this model be that if you would measure from these neurons, you should see also how the spatial temporal structure of this receptive field evolves? Right? So now you gave us one cross-section at about, 40 milliseconds after stimulus onset.
Yeah.
Right? But there must be physiology showing how this spatial temperature is evolving. Okay, so you're going to show us that?
Yeah.
That's great.
Thanks for asking. Okay, but are there any questions from the students?
Look, it's very easy, right? Because it just means you have to listen to our questions all the time, and it might get really annoying.
So you have to go through the one brain, one question.
Yes. So the backup solution that we employ in the lab is one brain, one question, which means every brain in the room has to pose at least one question during a talk. But I hope we won't get to that point.
Okay. Well, if you have a question, don't hesitate to interrupt. Okay. So, all right, so we computed this optimal linear filter, just as I explained before, and we can compare it to the actual measurements. And here's an experiment. This one is from Lesica et al., recordings from cat LGN, measured in response to the same natural movie that we use to generate this linear filter. And as you can see, the shape of the two filters is very similar. It has a biphasic response, okay? And the time component here, it's time is not stretchable because it's the same movie with the same frame rate used here and here. So this is absolute measurement, and as you can see, it agrees reasonably well. The spatial component, you can also see that there is center and surround, and the center is biphasic. The surround, not as much. And you can also see that the surround is delayed relative to the center. And all of this is reproduced by this optimal linear filter.
Just a quick question. In their experimental data, the surround seems remarkably symmetrical. I mean, are they averaging left and right?
Yes, they are. It's a circular averaging.
So they're averaging. Yeah. Okay.
Yeah.
And that's the membrane potential of the cell.
No, this is based on the firing rate. This is reverse correlation based on the firing. Okay. And just to remind you again, this was obtained with no free parameters. If you give me signal to noise ratio and the natural stimulus, there is a unique answer. So given that, it seems like a reasonable agreement. I should also say again, general theory should apply between species. This is a receptive field measured, spatio-temporal receptive field measured in locust. Again, in those neurons, L1 or L2. And you can see that the central part has this biphasic structure, as we know, but also they measured the surround, and it's a little bit bigger than in the cat. But I think that this is because of the stimulus that they used, which was concentric annula presented to actually emphasize the surround. So I think that this is not inconsistent with the predictions of the predictive coding theory. Okay. Now, if this is true, as Maniam said, the filter should change in response to the stimulus ensemble. And I have to say that there is some preliminary evidence that it changes in the way that is expected by this theory, but a more exhaustive study is clearly needed, and we're working to establish a closer collaboration with experimentalists to test this carefully. But what I can say now is that these people have changed the contrast of the stimulus just like was done in fly, and they saw that the response of the center gets longer and slower at low contrast. And in fact, that is, of course, well, I'm sorry, that's what they saw, that the response was longer. And this is the prediction of the predictive coding theory in the form that I described. Okay?
So by noise, do you mean photoreceptor noise or noise in the input stimulus? Because if it is noise in the visual scene, I don't know how the biological system knows what is signal and what is noise.
So again, this is the same trick that was used in fly, which is if you just reduce the contrast, okay, relative to the background
so it is intrinsic
intensity.
Noise in the system.
No, I think it's like a photon shot noise or something like that.
But I guess what Mr. is asking is that whether Partha, sorry, is asking is that you're not externally imposing more noise. It's all intrinsic noise, well, photon noise or intrinsic neural noise.
Yeah, that's right. If you presented, so that's a very good point. If you presented a noisy stimulus, and they actually did, you can present a white noise stimulus. How would the system know that it's noise rather than stimulus?
If it's spatio-temporal noise that is superimposing on, for example, a static scene, then maybe the visual system has some way of working out what the noise is and adapting to it.
Yeah, it could have some
cool.
prior.
That'd be really cool. That'd be really cool.
Yeah, it could have some prior. I don't know, but that's why I'm saying that has to be explored more in collaboration between theory and experiment. I think. Okay, so then what this tells you is that this predictive coding framework can account not just for the temporal aspects of the receptive field, but also for the whole spatio-temporal response of the system. And it doesn't just work in one species, but I think it works across species. So there is a fair evidence that this is the right way to think about the system. And in fact, other people thought that as well. And so I should compare with what has been done before. I'll do it very briefly. I can't do full justice to that work. But just to make sure we make a proper connection, there is one thing I should say. If predictive coding is done optimally, and the filter can be infinitely long in time, using signal values infinitely far back in the past to predict the current value of the signal, then you can prove a theorem that the outgoing signal, which is the prediction error, is white. This is the so-called orthogonality condition for optimal linear filters. Okay? So what this system does, it actually whitens the input and transmits a white signal downwards. Okay? The reason I brought it up is because other works, such as that of Attic and Van Hateren, used information theoretical approach to understand this, which actually treats the communication noise more explicitly. But, well, actually, Attic moved from that objective of maximizing information transmission to just the whitening of the signal by the early visual system. So whitening was actually his objective. And this is actually then related to the predictive coding theory, because optimal predictive filter whitens the signal. So what is the difference? Well, one difference is that if you start with whitening, you kind of push to work in the Fourier space, right? Because when you say the signal is white, it means that its power spectrum is uniform in the Fourier domain, okay? And so you compute the optimal filter from the power spectrum of the natural stimulus and the whitening requirement, but then you have to go to real space to come up with those spatio-temporal receptive fields that I showed. If you want to do that, just knowing the power spectrum and the Fourier domain is not enough. You also need to know the phases of the components, and that they don't know. So that was always an issue in Attic's and Van Hateren's work, and they used tricks to circumvent that problem. For example, in time, they used a minimum phase assumption on the filter. So for those who don't know what it is, don't worry about it, okay? It's a way to choose one particular filter out of the family of filters that have the same power spectrum, okay? And it's a very reasonable one. And in fact, it turns out that the predictive coding filter is by design minimum phase. But we didn't have to make this assumption. And in space, they usually use the circular symmetry assumption, which is also reasonable. But we don't have to rely on it. In fact, our filter will only be circular symmetric if the natural ensemble we use has a circular symmetry. If it doesn't, then we'll get something else. So we feel that our approach is a little bit more direct. It's in real space. And it also doesn't have those assumptions used in this way. And it's a little bit more explicit. Okay? Nevertheless, since their predictions were in Fourier space, let's go to Fourier space and compare what different theories predict and what the experiment shows. So these are the measurement of the receptive fields and the LGN converted into the Fourier space, temporal frequency, spatial frequency. This is what our kind of theory gives. And these are the predictions of Van Hateren and Attic as computed based on what they said. So we like to think that we get a closer shape to what is actually observed. But of course, one can say, well, in response, they could have tweaked those parameters. Well, they're not active in the field anymore, so they cannot tweak those parameters. So we are tweaking them now. And we are trying to get their theories as close to the reality as possible. And that work is still in progress.
But there's an important conceptual difference between the way I think Attic was approaching the problem And I always had difficulty with this notion that one has to maximize mutual information between the spike rate and the stimulus, because it made an assumption that there was a communication channel coming in from the outside world. And that communication channel, for it to be a sensible engineering concept, would have to include some kind of coding going on in the outside world that the system, visual system, was dealing with. But as far as I know, the outside world isn't trying to talk to us through some kind of code. And so that never made sense to engineering sense to me.
Maybe explain that problem a bit more detail.
Well,
I mean, maybe can we leave this for the discussion?
Yeah.
Because I think this is still an interesting question that we can discuss.
It seems like the predictive coding doesn't have the, has a sort of, it's a different engineering problem that is being solved than in the channel capacity problem. And the fact that there is a superficial resemblance may belie the fact that there is a fundamental difference in the way the problems are being posed, and that difference is important.
I think I think you're correct, and I'm happy to discuss this relationship more maybe after the talk. What turned out for us is that thinking about the system from the predictive coding perspective rather than information maximization perspective led us in the direction that I will show in the few more slides that if I have more time, and allowed us to make further predictions and understand the system better. And I have no idea of how people would have come up with that from the information maximization framework. So for us, it certainly was a better framework.
Yeah, I mean, I want to make a point here because we are talking about engineering principles. It's important to apply the right engineering principles because it's all too convenient to pick something like channel capacity and compute it without understanding why one is doing it.
So, are you going to tell us what the right principles are, Partha?
Well, I think
he hired me to do that. Okay, so now we're done with the first part, which was a normative theory of the spatio-temporal receptive fields. And hopefully I convinced that there is pretty reasonable evidence to think that the predictive coding framework is a good one to understand the system property. But what I'm going to do next is to say, well, not only can it be used to construct a normative theory, but it can also help to understand circuit computations on the level of individual neurons in the system and how they're connected, and mechanistically explain the appearance of that spatio-temporal receptive field. Okay? So the starting point for this direction is a very simple observation that if you look at the theoretically, this is actually the experimentally measured receptive field, it can be very temporally extended. Okay, of course it depends on the color scheme used here, but you can easily have traces going beyond 100 milliseconds in the past. So that means that the system somehow remembers what happened long ago. But neurons, individual neurons themselves, have pretty short time constants generally, okay? Maybe 20 milliseconds, 15 milliseconds. And even if you had a neuron with a long time constant, you don't just want to store a single value, right? It's an online problem. There is an ongoing stream that has to be constantly predicted and predictions being subtracted, and you have to remember what happened in the past. So how can actual biological neurons store this information for a long time, yet respond to quickly changing stimulus? Okay? So if this is the prediction error filter you want to have from the normative theory, the optimal one, this would imply that you would have elements with increasing and more increasing delays going back in the past that store information from the past or signals from the past multiplied by the weights as specified here and subtracted. Okay? And a neuron it would be hard to do this with a single neuron because it would have to be very slow. So how can we do it? Well, it turns out that we can modify this scheme somewhat by stacking up delays in the neurons in the stage-wise fashion. so that you produce the component of the prediction with the unit delay with a single neuron here, for example, multiplied by some weight and subtract. But you also send that delayed signal through the second neuron, also with a small delay. But now the total delay is the sum of the two, and that should be multiplied by the coefficient W2 specified by this, and so on and so on. Okay? So there is a way to use components with a short time delay, like actual biological neurons, to stack together to produce long delays. And in fact, I think the scheme has some value to it, because it suggests that the visual system might have two pathways running in parallel, okay? And the subtraction of the prediction happens in stages. You can think of those stages as the retina and the LGN, which is neat. But I can also identify those two pathways perhaps with the two major transmission channels known in flight, which are L1 and L2. Of course, this is just hand-waving and totally unsubstantiated at this point.
But do these, when you stack your neurons, do they need to be independent in this way? Like, you could also think about it in terms of this reservoir computing idea, where you just have densely recurrently coupled populations of neurons, and they would effectively be able to render something similar to this.
They could, of course, but so for example, in the vertebrate system, we know from the anatomy it's strictly feed-forward. There is nothing going back to the retina. That's why there is this.
No, it's in local circuits.
Oh, within that? Yeah, yeah, of course. Yeah, I mean, yeah. And we'll see some of that. Okay. But there is a problem with this scheme, because it kind of defeats the original motivation for predictive coding, which was reducing dynamic range. Because it is true that in this pathway, the dynamic range gets subsequently reduced at every stage by subtracting that correlated part. But in the other pathway, this is the intact original signal just being delayed more and more and more. So you still have to transmit the original signal. Then why do predictive coding at all? Of course, in the end, if you just transmit this, you're fine. But if you're using this model for retina and LGN, it doesn't make much sense. Well, it turns out that you can still save this idea. And you can save it by using a design that comes from engineering. Has all been invented, it turns out. Okay, so if I can get this. Okay, and there is a device called the lattice filter, which is used a lot in audio and speech processing, okay? And basically, you know, there are books written about it, but just briefly to introduce it, the way it's built, it's the same idea of stage-wise subtraction of the prediction. But instead of subtracting the prediction from this pathway only, you also do the reverse. And you subtract this part as a prediction for this signal. And by doing so, you reduce the dynamic range, not just in this pathway, but also in the second pathway. And so this circuit has all the requirements. It uses short time delay components to eventually take care of long correlations. It does so in a stage-wise manner. It reduces the dynamic range, and it has two pathways which are reminiscent of L1 and L2 in the fly. Please.
Earlier on, you said that the number of neurons is a problem because your optical nerve is just like.
Right.
Where do you put all these neurons?
Right. So, exactly. So the problem with this system is that it is indeed redundant. You've got to have twice as many neurons as optimally possible if you can build that optimal filter in one shot. Okay? It is true, we have to give something back. There is no free lunch. But the evidence that I'm going to show you in a minute hopefully will convince you that this is a reasonable model, at least a useful one.
So, just a quick question. When you say L1 and L2,
yeah.
When you say L1 and L2, which part of that circuit pertains to L1 and which part to L2?
I will talk more about this. But just to answer you briefly, we think that L1 is this part and L2 is that part.
Oh, I see. So, but as far as I know, L1 and L2 show very similar response characteristics.
Get to that too.
Okay. Okay. Okay, thank you.
Okay, so this is what's called the lattice filter, and before going too much in its many wonderful properties, let me give you two main predictions that this model would give you about the receptive field. Well, first of all, this is this prediction error filter as measured at this point. And as you can see, it has this familiar now form of the unit of biphasic response with the unit weight here and the weight less than unity here as before. And then this is the prediction error filter. The next stage, it uses an additional delayed path. And that's why its second phase is more extended and smaller in amplitude. So you would expect that when going from the retina to LGN, the receptive field would be temporally more extended. That's one prediction. And the other one is that in addition to this extension of the receptive fields, you also have the second pathways, and it's called the backward prediction error filter. And it's something not very intuitive, but you can think of it as predicting the value of a past signal based on the values that came after it. So it's not really prediction, but it's more of a postdiction. It's kind of has been invented not just by theorists like us, but used in engineering. Okay, and so there is a unit amplitude corresponding to the past, and smaller weights following it. Okay, so that's the second prediction. Well, is that really true? So do the receptive fields get longer when you go from the retina to LGN? And in fact, they do. Of course, this is a postdiction, just like backward prediction. And it has been measured by using this very neat technique. When you record from an LGN cell, you can identify not just the spikes of that cell, but also of the spike of a very strongly coupled presynaptic retinal ganglion cell. And by cross-correlating this with the input stimulus, you can compute by reverse correlation the filter corresponding to both the LGN cell and the retinal ganglion cell feeding onto it. And in fact, the filter of the LGN cell is more extended, as you would expect from this, than the filter of the corresponding retinal ganglion cell.
What about the reversed filters?
What about the reverse filters? So it turns out that this is also a postdiction. Okay, and it has been recorded, for example, in this nice work by Wolf and Palmer, where they recorded LGN temporal receptive fields of LGN neurons. It turns out that they are, of course, biphasic, but there are several kinds, and each curve actually corresponds to an individual neuron. And this is the kind that I would argue corresponds to the forward error like this one, and this corresponds to the backward error like that one. Now, the sign of the response is not important. You can turn it around, you know, switch between on and off cells. That's not important. What is important is the relative weight or area under the curve for the first phase versus the second. Here, the area under the first curve is bigger than the second, as you would expect from the forward filter. But here, the area under the second phase is greater than under the first, as expected for the backward filter. Please.
Would this differentiation between pre and post prediction filters all correspond to the center on, surround off, and center off, surround on neuron?
So I think this is a separate distinction. But because the definitions are not standardized across labs, sometimes people confuse. And so I argue that you can have this filter both in the form where the first response is positive and the second is negative, and in the opposite one. It will still be the forward. Okay? What's important is the weight. Okay? And if one was on, then the other would be off. So on and off is a different classification from this one. Okay? But it does get, we can talk afterwards. People sometimes confuse. Okay? All right? So, yeah. So this is the prediction. Seems to work in terms of the weight. But if you look carefully, of course, there is this problem with the widths of the peaks like we had in fly. And in fly, we explained it by the denoising filter convolved with the predictive coding filter that will give finite width to this first phase. But the problem is more severe here. You see, the prediction of this theory is that the second peak is actually very thin, but in reality, it's very wide. And it drove me nuts for a couple of months. And it seems to work so well, but this is just a major discrepancy I couldn't live with. Well, it turned out that this was discrete time theory. If you actually try to build a continuous time lattice filter, you have to alter it somewhat. And as a result of that alteration, you reproduce exactly that width of the second phase. And the way you do it is instead of using delays in the second pathway directly, you do something very tricky. You put a delay in the positive feedback of the incoming stage, and this would be presumably in the photoreceptor. This is this low-pass filtering of the photoreceptor. And then in every stage, instead of putting feedback directly in the backward pathway, you put something which is known in engineering as an all-pass filter. It passes all the frequencies with the same amplitude, but it shifts their phases differently. So in some sense, it's a trickier version of delay. And that's what you need to do to take this framework to the continuous time description. And then the filters that you get are very similar to the ones that have been measured. So this is the theoretical filters, and these are the experimental ones. This is the ones that show the growing temporal extent from retina to LGN. And this is the prediction for the backward filter in the continuous time framework. You can see now you get a wide second phase, just like has been measured by Wolf and Palmer.
But isn't there a risk now you're overfitting your data? Because it's quite some stuff you start to add now, right? To just get...
I don't know of another way to implement the continuous time.
I understand.
Yeah.
But in terms of parsimony, is this the way to go? Is this now not sort of a challenge to a model of this kind?
There always is. We always have to be careful. But I think the way parsimony comes in, if you have multiple models that make the same predictions, but one has more assumptions than others. So if there is another model that we can compare it to, we can discuss which one is more parsimonious. To the best of my knowledge, this is the only model that predicts this backward error filters that have been observed. It's true, it's a post-diction. Okay?
So Dmitri, what would be an example of and would it be a good example of an all-pass filter? Would it be a pure time delay, for example, which transmits all frequencies at the same amplitude but with different phase?
Pure time delay is a special case of an all-pass filter. So in fact, we can talk about this later. I didn't want to go into this detail. There are weights on this two links. And if that weight is 0, you get back the delay. But this is a more general version that goes to continuous.
I see. Okay.
Yeah.
Thanks.
Okay. So are there any more things that are kind of wilder, a little bit wilder, to kind of appease Paul? Indeed, if you measured a prediction error filter here, it will have this funny triphasic shape. That's just what comes out of this all-pass filter. But again, it turns out that people kind of saw it, and this is so-called lagged cells in the LGN that have been discovered by Mastronardi. And again, there is no universal and standard definition of lagged cells in the LGN. So different labs use different definitions. Everyone would call this kind of cell with this triphasic response as a lagged cell. Whether this one is called lagged or not depends which lab you talk to. Okay? So this lab, for example, that has also measured receptive fields in the cat LGN, they call this lagged, this cell's lagged, and I think this has this triphasic structure that I'm talking about, and these are non-lagged. This is just regular forward prediction filters. In addition, they also mapped out the spatial receptive fields here, which can be compared to our prediction. And in fact, that works pretty well, in terms of producing more predictions and so on. Okay? So, as we always did before, if there is a prediction and the theory is general enough, it should work across species. So let's go back to flies. And what I think is happening there is that the early part of the visual pathway is also described by the lattice filter. But there are good news and the bad news. And the good news is that in the fly, a little bit more is known about the circuitry because of the work of other labs, but also my group has a big connectomics effort devoting to figure out the circuitry of the early visual pathway of the flies. We actually know much more of the connectivity than is known in the vertebrate pathway. So we can actually compare. And the bad news is there is very little physiology, and physiology is very hard. So we don't know that part. Okay, but let's try to compare what we can. And so in fly, photoreceptors synapse on these two cells, L1 and L2, from which you can record. And the recordings that I showed are indistinguishable between L1 and L2, as Antje pointed out. So can they still correspond to the forward and backward filter? And I think they can. And you can see it in two ways. One, these are recordings in response to the off-axis stimulus. And you can see that the second phase is bigger than the first in that case, for L2, but not L1. And I think this is the indication of the same effect, but now it's an interchannel coupling that goes through the cell called L4. And we can discuss in detail how that works, but I think this is at least a flavor of what I'm talking about. Okay? The other proof, I actually don't have a figure in this talk, but I'll pull it up just to show you that makes sense. Now, in terms of the circuitry which we have, so what happens is this part was, of course, known that at 1 LL2 gets synapses from photoreceptors, the low pass filtering in photoreceptors. It hasn't been known until recently that L1, L2 are strongly coupled by gap junctions. It has been shown by the Borst lab that there is this coupling. And what we are suggesting, it may represent this link.
So has anyone tried to electrically stimulate L1, for example, and show that you're going to respond in L2? And
yes.
Vice versa?
Yes. They've shown it by propagating label through the gap junctions, and also by showing
that's showing it anatomically, though.
Anatomically,
and also physiologically, I mean.
Yeah, and also what they did was they had a genetically engineered fly where they knocked out transmission from L2 downwards with Shibiri.
Okay.
But
and that affects the response of L1.
Yeah, but the L1 response was fine.
Oh, that means they're not coupled. Oh, is that right? If you alter the response of L2 and the response of L1 remains unaltered, it means they're not coupled, right?
Right.
Electrically.
But when they then used the potassium channel KIR1 something to shut down L2, they were able to shut down the system.
L1 as well.
And yes, and they used that argument as an evidence for the gap junction. So if you just block the transmission out of L2, this part of the system works correctly. But if you electrophysiologically shut down L2, then this system is shut down too. That was their evidence.
Yeah, but in your case, they shouldn't only be coupled. They should also have, let's say, opposite filtering properties.
Right. So how does that come about?
Right.
I think that L4 is contributing to the story, which is, it's kind of like this, but not that exactly. And there is a scheme in signal processing that does this by another channel like L4. So it is possible. We can talk afterwards. It gets a little bit heavy on math. But in the end, the best way to address those concerns is to show you something that I didn't have in this talk. But I'll... no, that's not it. Sorry. So I'm just scrolling down to the correct figure. Here it is. So this is a predicted response of the forward and backward pathway in the lattice filter to a step stimulus. So this is the traditional prediction error filter, except that it's flipped because we think that the response of L1 is minus forward error, okay? Because in flies, everything is reversed. Or rather, I should say, the photoreceptor to LMC synapse is sign inverting, okay? And, but the L2 response, we think, is just the backward error. The difference between the two is not just the value of this peak, but the steady state part of the response. It has different sign relative to the transient. That's the reflection of the second phase being stronger than the first. And these are calcium imaging experiments that recorded signal from the axonic terminals of L1 and L2, as opposed to electrophysiological recordings which were done from the cell body. And when you record from the axonal terminals, you see, in fact, this structure that the lattice filter predicts, which is this part of the response has the same sign as the transient here. It's below the rest state. And here it is opposite, so it's positive, while the transient is negative. So that's the best evidence I know at this point. Of course, it's just calcium imaging, so you have to take it with a grain of salt. But I think that the way those cells work, there is a major difference electrophysiologically from what you can see in the soma and what you can see in the axonal terminals. Does that answer your question?
Yes, I think so.
Okay.
As far as I can tell.
Okay. That's important. I don't
No, but to understand the calcium imaging,
yeah,
what you're imaging here is the output of L1, L2.
That's right.
Right?
That's right.
Well, the filter is supposed to sit at the input side of it.
No. So, see, let's go back to this. So I would say that the output of L1 and L2 is this point. Okay. That's the projection to the medulla. Mm-hmm. Okay? And so this kind of computation happens in the lamina. And the filter is, but the filter is recorded in the output terminals. Okay. And the recording of the soma is probably more indicative of what happens right there. But, I don't know. Okay? So what happens afterwards? Can we see the second stage of the lattice filter? And that's actually where our connectomics effort is aimed at. And I would have to say that we're still looking at it. There are some similarities, like there is this branching, which could be corresponding to that. And actually, it may be, actually we haven't been focusing on this aspect of the processing because the most famous established problem in fly vision is that of motion detection. So instead, we actually focused on motion detection that happens in this stage. And interestingly, one can think about motion detection also in the predictive coding framework, because if you can detect motion, it will improve the quality of your prediction. By knowing that an object moves, you can make a prediction better. But the prediction has to be nonlinear, because you have the same signal in response to a white edge moving from left to right as a black edge moving from left to right. You can't understand it with the linear filter. You have to add a nonlinearity. And that's what we're working on now. And I don't think I should go into it. I should just finish up. So what I'll do, I'll mention the acknowledgements and then show you the movie of the reconstruction that we have. So hopefully I convinced you that the predictive coding framework is a reasonable normative theory that makes a lot of predictions from very few assumptions, which is what we like. In our hands, thinking about predictive code, about the system from predictive coding perspective, led us in the direction of this lattice filter model, especially knowing about the L1, L2 pathways in flies. That's what really pushed me in that direction. But I think it also applies well to the vertebrate visual system and explains some of the electrophysiology that has been done there. And finally, this is the last part that I'm not going to go in, about the connectomics, and you will hear about this from Moritz, I'm sure. Just to thank the people who contribute to this line of the work in the lab. These are the people who are working on the theory, and these are the people that I work with on the connectomics that I haven't talked about, but I want to show you the movie. So what I'm going to show you now is the cartoon visualization of our reconstruction project, where we take this electron microscopy data set from a cube of tissue like this one. This is from the fly visual system, and it's in the medulla, this is the second neuropil, and gets inputs from the retina called R7, R8, the color photoreceptors, and this LMC cells like L1, L2, and so on. And these are the ones that you're seeing here. And this is the size of our reconstruction. It's about 37 by 37 by 50 microns. It's very small, but it already contains more than a single processing column in the medulla. And so now I whiten all the inputs, and in color, I'm adding postsynaptic terminals to those neurons, L1 and L2. And these are neurons that have names, very creative names, starting with TM, MI, and DM, and so on. They all have some meaning. Nevertheless, we were able to reconstruct them for the first time through electron microscopy and identify them by their shape by matching to light microscopy, which was previously done. But the advantage of electron microscopy is that we don't just see all those shapes. And we can, in addition, we can also see the synaptic connections between those neurons. And there are about several tens of thousands of synapses in this volume. And we've traced a large fraction of them. And we were able to determine which neurons connect to which by how many synapses. And so we were able to come up with a circuit that describes this part of processing. And I keep adding more and more neurons in this volume that we have reconstructed. So now I've added neurons from neighboring columns. And there are also neurons called tangential neurons, which run this way as opposed to this way, and they're added now. Can we turn off the light?
Yeah. The property of this all-pass filter?
Right. So, so in the discrete time, what you do is you just have a unit delay in the simplest formulation. You have a unit delay, At every stage, you extend the length of the filter by that unit delay. So at every stage, you can get a longer and longer predictive coding filter. Okay? So the more stages you have, the longer you go back in time. So in, for example, audio speech processing, they use hundreds of stages of the lattice filter to do a good prediction. Okay? Now, when we switch to this continuous time framework, what happens is that it uses another basis to represent the signal other than just from the shift operator from the time delay. That other basis that is formed by Laguerre polynomials allows you to represent long correlations with the fewer basis functions, because each basis function has a long temporal extent. So when you have one stage, it doesn't just describe a unit delay, but it can be chosen to describe a delay corresponding to the width of the autocorrelation of the signal. The second stage, like, say, in the Fourier decomposition, can look at more precise shape of that, of that response.
So what would change from the first to second stage is the resolution, more or less, of this function, so the precision of this function.
It is both the resolution and the spatial extent. So they also get, unlike the Fourier harmonics, where you define the interval, and on that interval you just increase the resolution, the Laguerre polynomials do both at the same time. They increase the spatial resolution, but they also get a little bit longer.
So like in wavelet decomposition?
Like in the wavelet decomposition.
Okay, thanks.
Other questions?
So it's really interesting, the lattice filters. I think one of the properties they have is they are relatively insensitive to round-off error when you implement them in finite precision arithmetic. And that's probably related to the dynamic range issue.
Exactly, exactly. So there are many very nice properties. One of them is that they're insensitive to the, or less sensitive to the rounding off errors. Also, they do much better for ill-conditioned problems. So when there are strong correlations in the input, they do better than just finding a single linear filter. It's like when you invert a matrix, you have to use numerical procedures that are better controlled. And then one of the other interesting observations is there are these weight coefficients for the crosslinks that have to be computed instead of W's. For the tap filter, they have to be computed for the lattice filter. And the way they can be computed for the lattice filter is just based on the local activity of pre- and postsynaptic neurons according to the Hebbian rule. So they're naturally designed, it seems, to be implemented by neurons.
My other question was, you showed the spatio-temporal response of the filters you designed and the experimental data. To actually see what that does, wouldn't you want to run a movie through that filter and see what the result is?
Of course.
What would it look like?
So that's, I mean, I don't have a movie, but let's see, I have a single frame. So that's what a single frame looks like.
But since it's a spatio-temporal system,
yeah,
to really see it in action, you'd really want to run a movie.
I don't have it with me. Yeah. Yes.
But I'm wondering, because it kind of looks like a moving edge detector when you look.
Oh yeah, of course it's an edge detector, both in time and in space, yes.
But a moving one, so maybe it selects for certain velocities or.
Yes, but it's linear, so it cannot have preference for a particular direction.
It's isotropic, yes.
That's right.
So where does L3 fit into all of this?
Just say I don't know. Is that allowed, partner?
Any thoughts or?
Okay. Any other questions?
Yeah. Yeah, I was just rehearsing it there in the hallway. Sorry. But so we start with the whole issue of signal transduction and its problems, right? And from there, initially the inspiration was, okay, you know, there's these predictive filters and they're great because they solve all these channel problems. And then in the end, we forgot about the channel problems and became an optimal filter, and that's all great. And then it's really wonderful, right? How you show that this really maps to some of these receptive field properties that you find. But now, so if you look at this whole analysis of this L1, L2 system, at some point, also the fly shouldn't be worrying too much anymore about how to optimize its signal transduction. It has to say, no, now I need a neuron that gives me a response that makes sense behaviorally. As for instance, now let's take an LGMD neuron or something, which would be a very clear example of that, right? Or a neuron that just gives you a clear response with respect to some optic flow properties of the world, right? So now, so then we had this decoding issue with respect to function, right? So how are you going to solve that now? Because at some point we just have to switch gears. We said, okay, fine, now we optimize our signal, but now you, for your L1, L2 system, you have one step left. You have one synaptic step left to give you a functional, meaningful response. So how are we going to do that?
Right. So I think this is a great question, and we did start, as I said before, we started this motivated by the dynamic range reduction and the compression algorithm. And I think this actually kind of flies in the vertebrate pathway, just because of the sheer number of fibers you would have to have. But in fly, it doesn't really work. There is no clear bottleneck. And also in the vertebrate visual pathway, there is an expansion in V1, so it can't be the whole story. So what I think is happening is that the idea of decorrelation or stage-wide decorrelation of the input is a computational algorithm to generate features that can be used to learn the correct responses. How is that done? Again, inspiration comes from adaptive signal processing. And the way they do it is they build a lattice filter like this. And then if you tap that filter at this point, and also combine it with the original signal, you get an orthogonal set of features that are extremely good for using to predict another channel of side information. Okay. So for example, if you want to learn a certain response or you want to learn an association, the best way to do it, it turns out, is to decompose your input into those features that are the outputs of subsequent stages of the lattice filter, because they happen to be orthogonal. Mm-hmm. And this is good.
So they're saying the principle, in some sense, is to get to decorrelated and maybe maximally sparse responses of your input space. Is that reasonable?
So sparsity we don't completely understand because that will involve nonlinearity. Mm-hmm. But low dynamic range, yes. Okay. So the number of spikes, at least in the vertebrate system, the number of spikes decreases at every stage. Mm-hmm. Yes, that's known. Okay? But moreover, those features, if you plot them in the correct space, they are orthogonal. Mm-hmm. And so there are an appropriate set of features to learn a response, which is the action that the animal has to perform.
Right.
I didn't know that. We came to the lattice filter from the perspective of minimizing dynamic range and decorrelation. But when I started reading about the lattice filter, it turns out that that's exactly how they're used, not just for decorrelation, but to generate those features that are put out here and used to predict another sequence, for example.
Right. So now, given that everybody had a big breakfast, so we have plenty of time to get to lunch. So in some sense, the coding model that you're presenting here is very much based around rates.
Yes.
But there's also this literature that talks about, let's say, a more temporal coding component already coming in at the retinal level, right? That ganglion cells give you a latency coding, right?
Yep.
And in some sense, you're not taking that on board in this model. Or maybe you do, but then you have to explain it to me. So my question is a little bit, you can see temporal coding as sort of an additional coding channel you have access to.
Yep.
Right? But you don't seem to really exploit that. Is that something in the future, or is it because you believe temporal coding doesn't matter?
If you give me another hour, I can tell you about
like I told you, we had a big breakfast, so we have plenty of time.
No, so in the parallel line, we have a parallel line of work where we're trying to understand the spiking as a case of analog to digital conversion in neurons. And again, inspirational comes from signal processing. But at this point, we just see it as an encoding of an analog signal in an efficient way, nothing more.
Okay. Very good.
Thank you. Yeah, I think let's thank Mitya, and we'll break for lunch. When should we reconvene?
The tutorial starts at 15:30. At 3:30.
3:30.
Okay, and it's in room 54.
5403. It's the building called Cahier, so follow the signs, and it's a room that has a big window overlooking the plaza, so you will find it. Very interesting.
So let's thank Mitya, break for lunch, and meet back at 3:30.
Thank you.
Very great. Good start.
Thank you.
Yeah.